target interaction prediction from chemical, genomic, and in an integrated framework. This is a joint work with and Susumu Goto. In this talk, I begin by providing research. After the preliminary analysis of interaction networks in our data, I will show you a method to predict interactions and some results. Most of the drugs which interact with their target proteins, biological behavior of the proteins. Identifications of the interactions between is an important issue in genomic drug discovery. Examples of pharmaceutically important target ion channels, nuclear receptors. The Genome Sequence Project revealed of these proteins. The same screening of chemical libraries for us to analyze the chemical space. However, our knowledge about the genomic is very limited. Special compound interactions. A drug-target interaction network can be represented by bipartite graph. So in this case, a blue node corresponds to what drug candidate was. A red node target proteins, target candidate proteins. From a statistical viewpoint, the problem of drug can be regarded as edges between using partially interaction information. To predict compound interaction, one popular method prediction is that similar chemical structure with the target proteins. With sequences are likely to ligand. Based on this discovery method or machine learning method, arguments are formulated of bipartite graph inference. Another approach is to use pharmacological information for the drugs. We call it a pharmacogenomic approach. For example, the side effect information proposed recently to predict a missing. The prediction is formed based on the assumptions that with similar effects such as draw synapse to share the same target protein. However, there is limitations on the applicability. The side effect information obtained from the drug. So the method is applicable to drugs. But it is not possible interactions between arriving compounds and the protein. I summarize two approaches. The chemical genomic approach investigates the relationship between the chemical space or possible structures and genomic space or possible genes or proteins. On the hand, the pharmacogenomic approach is to investigate the relationship between the pharmacological aspect of therapeutic effect, including side effect or pharmaceutical, and genetic space of possible genes or proteins. In this research, we try to combine these. First, we investigate the relationship between the pharmacological space and the topology of drug target. Next, we develop statistical methods to predict drug target interactions on a genome-wide based on chemogenomic and pharmacological data. First, we extract interaction from public databases, Genomic, SuperTarget, and Drug Bank. In study, we focused on drugs which target enzymes, ion channels, GPC, and nucleases because they are known to be used for drug development. The detailed statistics in this table used these data, standard data. Here we define pharmacological for drug. We extracted about 7 pharmacological effects from the drug package insert stored in the mining, and we represent by a binary profile like this. Pharmacological keyword is coded as one across the 13,000 keywords. Based on this process, we generate by using weighted coefficient. Here we because we want to put rare specific keywords and smaller common keywords. We compared pharmacological similarity with chemical structure similarity. Here we evaluate the chemical structure similarity, the graph format between the two-dimensional curves. And we compute the similarity scores based on the coefficient, which is defined as a rate of the size of common graph structures, the size of the structures. This figure shows a scatter plot of pharmacological similarity scores against chemical scores. You can see that they are correlated, but any exceptions. For example, these regions corresponds to high structure similarity, but low similarities. Next is the distributions of the similarities against network distance. By network distance, we mean the space between the drugs on the drug target network. Remember that the drug target network is by a Bipartite graph. So left figure shows plots of the distributions of chemical structure similarities. Shows the box of the pharmacological similarity distribution. You see that the distance, the lower the in both cases. By comparison, This tendency is much stronger in the case of pharmacological similarity. At the large, the rest of the similarity scores are 0, and the difference between the distance 2 distance 4 is very important because correspond to drug pairs which share at least one protein. On the other hand, the distance 4 drug pairs which do not share the same target protein. So we expect pharmacological information may be useful for drug actions, but the availability of pharmacological information is limited. We propose the following two steps. First, we predict a known pharmacological chemical structure. Next, a bipartite inference with pharmacological similarity and Mexican similarity for protein. In the first step, predicting unknown pharmacological information. These two matrices show situation in similarity matrix. We compute all. However, in the pharmacological similarity matrix, to predict this parts, we propose a regression model. Limitation, don't go into the technical details, that this model can learn the correlation similarity matrix in the pharmacological similarity matrix. In the second step, bipartite graph inference method. In most of the algorithms for a given compound protein pair is formulated like this. This is a rate of compound similarity function and the functions. They use pharmacogenomic functions for compounds. If the term score a threshold, we predict the compound to interact with each other. And we evaluate performance by performing. We split drugs into two parts: training set and test set. To model based on the training set only to the test set, and we evaluate the prediction accuracy. Evaluate all performance. We draw which functions of true positive rate based on threshold. Computing AUC score, and we tested structure to pharmacology and predict pharmacological similarity. You can see that the use of pharmacological outperform similarity. When we use the predicted version of pharmacology, we obtain the result is chemical similarity in most cases. Finally, we make comprehensive of unknown drug target interaction. As test commands in the cake ligand database, all uncharacterized drugs in cake. As test proteins, we used in the cake gene database. And we use data as a training in the running process. As a result, we focus on the 1,000 persons based on structure, chemical. We confirmed that 140 predicted pairs in the other protein ligand interaction data. When used a proposed method, about 100 predict in the other protein ligand interaction data. For we investigate the biological validity based on scientific literatures. We could confirm that some of the predictions to some wet lab experiment based on the research information. Some of this drug targeting correlate with pharmacological similarity than with chemical. Proposed method enables us not target interactions on a large scale. The method does not require 3D structure information, and is based on the integration of genomic and pharmacological spaces. I'd like to thank collaborators and my colleagues. Okay. Thank you for your attention.
Any questions? Go ahead to the mic.
I may have missed. How did you measure chemical similarity?
We evaluate the chemical structure similarity based on the graph alignment. We compare the
This is
graph structure.
This is 2D.
Yes, 2D graph structure. Yes.
The result difference was 3D.
We haven't tried yet.
One question. So maybe I missed the slide. So how much better when you add these pharma-related kernels on your original calculated kernel? How much better the performance?
In terms of the ROC scores?
Yeah.
Okay, I'll show you. How much the improved?
Okay.
When we use the true pharmacological similarities, the gain a much result compared with the chemical structure similarity.
So for this family, it has a really large improvement. Maybe.
No, I don't understand why this.
Thanks.
Okay,
Okay, thank you.
Any more questions? Sorry. So I have a comment or question. If I understand correctly your work, then what you try to do is to use pharmacogenomic or side effect information in order to predict drug targets.
Yes.
Now the question is, there are many properties of drugs you could use for predicting drug interactions. So you mentioned chemical similarity, you mentioned on ATC similarity, some other categorization. The question is, have you ever tried to just integrate them rather than compete one with the other? So why not somehow integrate them and maybe you predict better drug target interactions?
Yeah, I think it is very interesting research. Actually, I tried the integrations of chemical structure similarity and pharmacological similarity into a single one, and we applied by the method based on the integrated similarity scores. In my opinion, that procedure doesn't improve, so that's why I didn't present the result here.
Okay, so let's thank the speaker again.